I'm Matt Pillar, Chief Editor at Bioprocess Online and host of the Business of Biotech podcast. I'm thrilled you made the time to join us today. We have a lot to cover over the next 60 minutes, and we've assembled an awesome panel to cover it. Joining me today are Roberta Duncan, VP and mRNA Program Lead at Seqirus, Dr. Jin Zhou, Executive Director of Process Development for mRNA and Protein Biologics at Ultragenyx Pharmaceutical, and Dr. Jason Dickens, Associate Director of Upstream and Downstream Process Development at the Duke Institute for Human Vaccines. They're all on the front lines of the effort to leverage messenger RNA to prophylactic and therapeutic effect, and they've graciously agreed to spend the next 60 minutes with us to discuss their work and answer my questions and yours. On that note, as you ask questions of our panelists, I would just ask that in addition to using that Q&A box, that you address specific questions to specific panelists, if at all possible, so that they're best. So we choose the panelists who's best equipped to respond to your question. So Roberta, Jason, and Jen, I'm super honored and thrilled that you could join us today. Thank you for making the time.
Thank you for having us.
Thank you for the invite.
Yeah.
Thank you.
So I'd like to start with COVID because it's a likely place to start, right? mRNA vaccine development has been in the works for many, many years prior, but the pandemic shifted attention on this space into absolute overdrive. Nature just last month published an article that posited that at the end of 2019, so 2019, just a couple of years ago, the combined market cap of the 5 at that point publicly listed companies focusing on mRNA platforms, their combined market cap was $15 billion. As of August 2021, so just a few months ago, that cap was more than $300 billion. $300 billion. So the explosion is, it's unlike anything that we've probably seen in a long, long time. So Roberta, I'd like to begin with you again, starting with COVID. I look at Seqirus and I see sort of a classic example of an established leader in traditional influenza vaccines that's now forging into the mRNA space. Give us a bit of the backstory on Seqirus's foray into mRNA and how the company is building on its long history in flu protection and vaccine manufacturing with this newer technology.
Yeah, thanks for the opportunity to talk a little bit about Seqirus. We have a longstanding history in influenza vaccine and protection and prevention of influenza virus as a public health challenge. And while Seqirus formed in 2015, and we have taken the helm of being a flu-focused company, it's not new for CSL. So CSL has a 100-year-plus history in this space, and actually dates back to the Spanish flu. So we talk about in 1918, the 50 million plus that died, and we fast forward into 2009, where we had the H1N1 swine flu pandemic, right? So Seqirus and CSL have been in this space for a while. And the challenge that we are facing is how do we bring additional options and technology to the table? And mRNA presents with that opportunity. And so while this has been a really exciting time in our industry around how do we pull together the right resources and the brains to the table in order to deliver a COVID vaccine in this crisis that we are experiencing, been working on the next generation mRNA in our research team for a number of years. And so we're working on the self-amplifying mRNA, and there are differences between the traditional and self-amplifying, but we're not new in this space. And so we're continuing to forge forward, and we are looking to really go into the clinic next year with our mRNA vaccine focusing on flu. And so I guess your question is, what is some of the history that we bring to the table? Influenza manufacturing, influenza producing a vaccine that addresses 4 strains of influenza every year, twice a year, that requires quite a bit of technology, that requires quite a bit of proficiency and productivity. And so these are the areas that we continue to lean in very heavily. And when we look at COVID specifically, in Seqirus, we did some work on developing a COVID vaccine with the University of Queensland in Australia, and that did not progress forward. But we did also work very closely with AstraZeneca to manufacture doses of their COVID vaccine for Australia. So we've leaned into those areas of other technology that can that have infectious disease prevention and protection potential, and we're continuing to advance our own differentiated portfolio, which is focused at influenza.
Yeah. So let me ask you this: when you starting where you stood, what was necessary from an IP and capital or infrastructure perspective to sort of get in the mRNA game when you did?
Yeah, so we've been in mRNA for a number of years. And when we think about our investment in this space, our investment will continue to be around the core capabilities of understanding the technology. What we look at from a capital perspective with regard to our manufacturing footprint, we have a global manufacturing network today, and it's designed to be scalable to manufacture large volumes of vaccine. And it's also designed to be incremental. So we'll be building upon our capabilities to expand in mRNA. And so when you think about the footprint for mRNA, there is a lot of promise, and we're seeing some of that being proven in the manufacturing of mRNA today, is the footprint may not need to be quite as heavy from a capital perspective than you have with other manufacturing modalities. So that still needs to come to bear, and that's something we're looking at Seqirus very closely.
Very good. Okay, Dr. Dickens, I want to turn to you. So similarly, the Duke Human Vaccine Institute, you've been an academic leader in the development of a wide range of vaccines for a number of indications, from HIV-1 to tuberculosis, influenza, syphilis, gonorrhea, cytomegalovirus, rotavirus, parainfluenza, Zika flavivirus, and plague. I mean, it goes on and on. It's now added SARS-CoV-2 to the lineup, but the Institute is really interesting to me because you're more than just academic. You're actually producing clinical supply in a CGMP facility. So tell us a little bit more about the work that DVHI is doing.
So, excuse me, so our background is in mapping disease states, right? And back about 10 years ago, we used to outsource that ability to supply phase 1 clinical trials. And that became quite untenable, both from a time and space and cost perspective. At that time, they decided to bring in a process development capability and GMP capability to feed phase 1 and phase 2 clinical trials. So in the 10 years, they built out that capability to be able to service the needs of the researchers and driving into and translating that into clinical trial knowledge. So we've been quite successful at building out facilities and process development teams and skill sets to be able to deliver to that end.
When did the Institute begin working in mRNA?
We began to work on that in 2017, 2018. Because of the emerging viral disease, understandings of those complexities of the epitope and things of that nature necessitated multiple immunogen constructs, from protein trimers to nanoparticles and messenger RNA, of course. So it was at that time they decided to build out a messenger RNA capability. Of course, tangentially, pandemics require rapid response biotherapies, and certainly messenger RNA fits that bill, right? And we've seen that through the pandemic. So the other piece of it is relative to cellular processes. Messenger RNA provides a bit of that precise precision of expression of specific antigen, right? To induce an immune response. It's also flexible, where you can change the open frame readily to different constructs. And then it's also scalable, as we're seeing on the manufacturing space. And we certainly have a scalable platform as well.
Okay, we'll get into some detail there a little later on in the conversation. But I'm curious, in 2017, when you first entered the mRNA space, you talked about building out that capability back then. There was little to no sort of standard, right? There was not necessarily, not necessarily. There was no platform capability. So can you just tell us about just a high level, the evolution of mRNA production and manufacturing, what the environment looked like there.
So the environment really was limited in the raw material space. There was a lot of raw materials that weren't qualified for GMP. They also were not DNASE certified, some of the assemblies and things of that nature. So we spent a lot of time building out, the target raw materials, as well as the different assemblies and whatnot, make sure they could meet our needs, and developing processes when they couldn't be certified, developing processes to mitigate those risks. So we developed a platform that met those, borrowing from Drew Wiseman, who developed the non-replicating RNA, and we built upon that to build a platform that delivers consistently different types of messenger RNA, and I can walk you through that process.
Mm-hmm. Excellent. Yeah, we'll talk about that in a little bit here. Dr. Zhou, I'm thrilled you could join us because Ultragenyx brings a really unique perspective to the conversation. You're a representative of an innovative company that's leveraging mRNA for personalized therapeutic purposes beyond the current focus on vaccines. So welcome. Thank you for joining us. And why is mRNA fundamentally attractive to you as a rare disease treatment approach?
Ultragenyx is a rare disease company. So we develop a therapeutic for rare and ultra rare disease populations. And then some of the disease, we only have like a few hundred to a few thousand patients worldwide. So, in individual therapy, basically, you can eye it as a precision medicine for a small population. And in our portfolio, we have like small molecule, we have biologics, we have gene therapy. And then about 4 years ago, and the company started to invest in mRNA. The reason we chose mRNA is one, it can address, unmet medical need that other modality cannot. And especially when we talk about intracellular protein, when we talk about large molecules, and then in enzyme replacement therapy, mostly targeting a secretive protein, but a lot of target is actually intracellular. That's one. And two, for every single disease, almost we have to develop a unique process for that indication. That actually increased the development cost and development timeline. For mRNA technology, basically one of the unique feature is you can leverage platform technology to address different molecule. So, and certainly recently we developed GSD, a target, kind of like a transcript replacement therapy for glycogen disease, a storage disease. So for that one, it's traditionally that's a replacement therapy target. So we have a gene therapy platform. However, the gene therapy platform, the size of gene of interest is limited. It's only less than 4.5 kb in our platform. And for this particular mRNA or gene of interest is actually over 5 kb. So in that, it's also intracellular protein. So in that case, mRNA is the only opportunity to address this unmet medical need. That's how we started a program like 053 to target GSD3